Right, let's get started. So last time we talked about the immune system, and this week we have one more set of lectures, right, that are going to be on exam 3. So at the end of this week, that is when the content for exam 3 is going to cut off. Okay, and then we'll put the next week's lectures on the final. Okay, the final is not cumulative. Okay, so for exam 3, you just have to know this material. For the final, you'll just have to know next week and to the end of the course's material. Any questions? There'll be 7, so there's 7 lectures, I believe, to cover for exam 3. Expect even number of questions for each lecture. Focus on the lectures. Of course, it's always good to get a second opinion by reading the textbook, but I'm going to draw and compose questions that are based on lecture content. Okay, and I'll try to balance it out across every lecture, an equal amount for each lecture. So don't just spend, 80% of your time studying those first 3 lectures and don't get around to studying the last 4. Then you might, miss some important content. Okay. So today for the next 2 days, we're going to be talking about fluid and electrolyte balance and the process of excretion of things like waste and osmoregulation, balancing the fluid in your body, excreting particular ions, excreting particular dangerous things, right? And being able to maintain the overall level of osmolarity in your blood and your body's tissues when you're operating in sort of diverse environments that are sort of surrounded by fluids. And different animals are going to have different strategies for how they're going to be able to maintain that osmolarity. Like these marine birds will have little secretion areas on their beak where they'll be able to enter the waiting room. Where they will be able to excrete like salt at very high levels, very efficiently, right? Whereas we are not really in a salty environment all the time. We can use sort of much more, and we have different needs for what we have to excrete, and different demands on what we have to excrete. We're going to do that mostly through the filtration process of the kidneys. Okay, so let's take a look here. These are sort of the key concepts that you're going to want to know for the next 2 lectures. Why do we need to excrete things? And what are we excreting? Okay. What are the processes, the cellular processes and molecular processes by which we can actually selectively release material into our urine and excrete it? How does the volume of urine get created through the process of glomerular filtration? Then how does the sort of proto-urine travel down these tubules in the kidney, and different things are going to get pulled out or put into that urine in order to sort of perfect the excretion to get rid of only what we need and recover everything that we still, only what we don't need and recover what we still do need and do not want to have to gather from the environment. One of the sort of trickiest parts about this is going to be understanding the medullary osmotic gradient of the kidney. That is a sort of gradient of strength of salt and urea inside the kidney that goes from normally like isotonic to your blood to highly concentrated down at the core medulla of the kidney. And that gradient is absolutely essential for our ability to concentrate urine beyond the concentration of solutes in our blood. Okay. And that allows us in turn to sort of retain water, okay, in our blood when we aren't drinking enough water. And finally, we're going to understand a variety of different. Is that high-pitched sound? Yes. Is that from this? Yes. Oh, it's independent of this.
It's because the microphone's pointing right at the speaker, so it's
the mic's off though. So I think it's from other. I don't think it's. Yeah, it seems independent of that. That's super annoying though. Anybody else? I wonder where it's coming from. So I'm sorry about that high-pitched noise. It's not the mic. Maybe it's something else, but we'll try to solve it next time. Okay, so then we got to understand regulation of the kidney function. Okay, there's a couple different systems, our hormonal systems, and as well as sort of paracrine signals, the signals that are going to operate both on circulating compounds within the blood, pressure sensing changes in juxtamedullary apparatuses, things like this that are going to maintain the overall function of the kidney and be able to turn up or turn down how much water are you excreting, how many solutes and which solutes are you taking back up. Okay, so let's. Okay, so what is the overall function of the human excretory system? Okay, the first 3 things that are going to be broken are all part of the overall process of osmoregulation, which is really just how much water is in your bodies. Okay, and what is the composition of the ions within that water? Okay, so and within your body fluids. So we want to maintain the osmolarity of bodily fluids in a fairly narrow range, around 300 millimoles or milliosmoles. Okay, and we want to have the appropriate concentration of ions like sodium, potassium, calcium, magnesium in your bloodstream. Okay, and beyond that, the human excretory system is also going to, through the process of regulating how much water is in the body, is then going to also maintain the volume of blood plasma and blood pressure. Right, the more water that you retain, the higher the blood volume you're going to tend to have, the higher the blood pressure. Okay. Then, so beyond this, so this is all sort of linking into the function of the cardiovascular system. And the kidneys, actually, the nephrologists at the hospital often work hand in hand with cardiologists, because if you do something to one of those systems, you're going to impact the other system. Okay. And then finally, we want to eliminate toxic things like drugs. okay, or other things that we consume that could be toxic, or breakdown products of our metabolism. And then we're also going to be able to secrete particular hormones and enzymes that will not only regulate the function of the kidney, but also regulate other functions, oftentimes of the cardiovascular system. So, for example, where does erythropoietin get produced? That's part of the function of the kidney that's going to drive the production of red blood cells and increase the oxygen carrying capacity of the blood. Also, a molecule called renin will regulate the overall level of sodium retention in the kidney, which then sort of is going to, in turn, sort of indirectly regulate plasma volume. Okay, so that's kind of a brief summary overview. Now, one of the things that we want to, what is the most important thing that we need to eliminate that we have great excess of all the time in our bodies? Okay, it is nitrogenous wastes. Okay, we are animals taking in nitrogen. Okay, through proteins, through DNA and RNA, anything that you eat. Right? You've got amino acids. You're getting 100 grams of protein a day or something, but in that process of breaking that protein down, turning it into muscle, turning it into energy, you're going to also have to eliminate the nitrogen containing amino group from those proteins. So whenever every amino acid in a protein, an amino acid chain, a peptide chain in a protein, is going to have these amino groups on there. So when you break them down and you undergo catabolism of a protein, these are going to go free. They have to move to the urea cycle, and they have to get excreted. Okay. We also have ATGC in our DNA, RNA, or U in our RNA. Those are highly nitrogenous bases. Okay. And when you break those down, you also need to excrete that excess nitrogen. So we've got a lot of nitrogens floating around. We've taken much more nitrogen than we actually need within our body. We have to get rid of that. So why do we have to get rid of it? It really comes down to the structure. The most important reason we have to get rid of it is the structure of this amino group. Okay. So when that is cleaved off, it becomes ammonia. Okay. NH3. Ammonia is a gas. Okay, it's actually a highly toxic gas. Okay, and you can use it in cleaning supplies or whatnot, but inside your body, you don't want it inside your body because it can combine with protons and turn into the ammonium ions. Okay, so if you combine with a proton, what are you doing? You're taking up a proton, you're taking a hydrogen ion, okay, and adding it to that. That's pulling the hydrogen ion out of solution. Okay, you pull the hydrogen ion out of solution, you're going to raise the pH. This is a pretty strong base. Okay? It is going to raise the pH of your body's tissues, and that if you throw the pH too far out, many of your proteins functions will no longer operate efficiently. Okay, so and one other thing about ammonia is that it's a gas, so it can actually diffuse from your blood into your interstitial fluid, and from your interstitial fluid through your cell membranes into your intracellular tissues. If it does that, turns into ammonium ion, you can be screwing up your pH not only outside of your cells, but also inside of your cells. So it's highly toxic. It also can sort of mimic potassium ions in some ion channels and screw up the function of those ion channels, which are very important for maintaining heartbeat, maintaining nervous function. And so we need to get rid of ammonia. Okay. Now, there's different ways that we can get rid of ammonia. Okay. If you are a fish, okay, and you have a ton of water, if you are a sort of a freshwater fish and you have a ton of water sitting around, you can just let ammonia diffuse out of your gills. Okay. It's very easy for you to get rid of ammonia. Okay. That's because the ammonia is very soluble in water, and the fish have a great excess of water around them. The freshwater fish are constantly trying to get rid of water that's seeping into their body. Okay, so it makes it very easy to get rid of ammonia. For us, okay, we need to try to remove ammonia in a more concentrated way. Okay, because we cannot allow the concentration of ammonia. We only need a few parts per million of ammonia before it starts to get toxic within our bodies. So we need to convert it into a substance which we can then which is much less toxic, which we can tolerate in a much higher concentration within our body. And So what we do is we are going to synthesize through the urea cycle, turn ammonia into urea through a chain of reactions, okay? And urea is still quite soluble in water, okay? You can have it still at a high concentration. And now the vast majority of how we are going to excrete ammonia is through urea. So it takes a moderate amount of energy in order to make urea from ammonia through that cycle. The toxicity is much less than ammonia. And it is going to require less water to excrete than ammonia because it's more concentrated. We've got 2 nitrogens on it instead of 1. So for terrestrial animals, most of our nitrogen excretion is through urea. Now you can also keep going and turn it into uric acid. This uric acid looks a lot like uracil actually, which is one of those RNA bases. It shares a similar sort of molecular structure. And uric acid is something that does form within our bodies, okay? If we have a high concentration of urea within us, okay? And if we have too high a concentration of uric acid, it actually causes problems and diseases. But in other creatures like birds, where they need to maximize the concentration of what they're excreting, okay, they're going to actually use uric acid for primary mechanism for getting rid of nitrogen. But uric acid is very insoluble in water. It takes a lot more energy to synthesize that complex molecule. It's quite non-toxic though, and it takes very little water to excrete it because it actually precipitates out of solution, and you can just actually expel it without any water at all. Okay, so I guess here's the summary of that, of ammonia. I've pretty much all said everything there. Submit that person, okay. Urea, also just said all of that. The person who originally synthesized, so the very first origin of organic chemistry in the early 1800s with Friedrich Wöhler synthesized urea from inorganic components. So why is that important? Okay, well, first it's interesting that we can sort of show that life is made up of components that you can piece together from other things that you found in nature that are non-living. And 2, the synthesis of urea was instrumental in the development of farming and the increase in production of farmland fertility, because you can use urea and other nitrogen products as the source of nitrogen for plants to grow. Plants need nitrogen in order to grow. So the ability to industrially manufacture urea sort of was part of the big agricultural revolution. Okay. Finally, just to look at uric acid here. What is all that white stuff that the puffins are gripping? What's the white stuff on your car when they're peeing on your car? Okay, it's just uric acid. The white stuff also that you can't even get it off the car. Why? Because it's not soluble, right? You got to scrape it off. Okay, so that is one of the advantages for the birds, and it's advantages for you for uric acid. Okay, so what happens if we have too much uric acid? Okay, the disease that tends to result, the most common disease is gout. So this is generally initially manifested as a buildup of uric acid crystals in the joints of the body. This causes an arthritis. Okay, it first generally is first felt in this joint of the big toe, and this was first observed. This is one of the first things that von Leeuwenhoek observed under his microscope that he developed, these little crystals of uric acid. Okay, so gout is caused by excess urea. And one of the reasons that it builds up is that even though we can produce uric acid, we lack an enzyme that effectively breaks uric acid back down into urea. So once it builds up, it's difficult for it to remove once it crystallizes. So what drives gout? Excess meat consumption, excess beer consumption, anything that has a lot of purines in it, it's nitrogenous compounds in a great excess will tend to exacerbate or initiate or exacerbate the presentation of gout. Okay, so that's sort of like a brief overview of what we're trying to get rid of, the nitrogenous waste. And now, what is the actual organ systems that are going to do that for us? Okay, so the key organ, and what we're going to spend most of our time talking about in this lecture and the next lecture, is the kidney. Okay, kidney's incredibly important organ, right? You need it for function. It's one of the most transplanted organs. Luckily, it's so important. It comes with two. Okay, you've got a backup. If one kidney goes out, you can still live off the function of your other kidney. Okay, that is how important it is. You only have one heart, got two kidneys as backup. Okay, so what does the kidney do here? And this is an interesting structure. We can sort of see basically the structure of where urine is produced and collected, as well as this capillary network surrounding that structure. And the kidney has a very interesting anatomical structure. Okay, in the structure of the kidney is intimately related to its function, and it cannot be separated from its function. Some organs, like the liver, are just a big lump of cells that are like a little factory. Each cell is its own little factory. It's converting things from one molecule to a different molecule, and the overall structure doesn't matter that much. Kidney, the structure is maybe the anatomy of the kidney is maybe the most related to its function out of any organ. Okay, so looking at the kidney, what does it have to do? Okay, it is involved in osmoregulation, and the three things you need to do for osmoregulation in both the cytoplasm that's inside your cells, your interstitial fluid, the fluid around your cells, and in the blood. So we need to regulate the overall volume of the blood in that fluid. Okay, we have to regulate what is dissolved in it, the solutes, and we have to be able to clear toxic things or metabolic byproducts out of there. Okay, so our main organ that is going to do that in humans is the kidney, and the kidney is broken down into a collection of parallel structures called nephrons. Okay. You've got about 1 million of them in each kidney, and those nephrons are going to work together to do a number of things. First, they're going to collect very large volumes of fluid from the blood. Okay, so like you've got 5 liters of blood in you. How much fluid are you going to collect of blood plasma? You're going to collect 180 liters per day on average. Okay, so that means somehow your blood, you're collecting, I don't know, 40 times the amount of fluid in your total fluid in your blood is going to actually get collected in the kidneys and somehow returned back to the blood during this process. So this is a non-selective process. We're going to get that fluid out of the blood, okay? And then you're going to modify that collected fluid, okay, by selectively excreting or secreting ions or small molecules into the fluid, okay? And then we're going to reabsorb other molecules that we want to retain and bring back into the blood, okay? Then return that to the circulation. And then finally, we need to get rid of the final highly modified product, the urine, okay, by the bladder. So let's look a little bit more into each one of these things. Volume regulation. How do we accomplish volume regulation sort of conceptually? Well, we basically have to change how much, at the end of the day, the volume regulation is all going to come down to how much you drink, how much fluid you consume, how much fluid you sweat off and evaporate off of you, and how much urine you produce. So you can produce a wide range of urines from less than 500 milliliters a day. If you're in a sort of hot desert environment, we haven't been drinking enough fluids, or you could go up to 2.5 or even more liters of urine produced a day, depending on how much you drink. So the kidney has a huge ability to vary the amount of urine that's excreted, which then allows you to maintain the overall amount of fluid within your body. So that's your volume regulation. And we also have solute regulation. So the urine can go from hypoosmotic, that is to say, the concentration of solutes that is much lower than in your blood or in your interstitial fluid. So your blood interstitial fluid is around 300 milliosmoles. You can make that 3 to 6 times more dilute than the blood interstitial fluid in the urine. But we also can change that up to a hyperosmotic concentration, up to 4 times more concentrated than the blood. And that sort of change in the amount of solute is intimately related to the amount of volume that we're going to be producing, right? So if we are producing much more urine, that's generally going to be a hypoosmotic urine, right? If we have much little urine, a small amount of urine, it's probably because it's hyperosmotic. We're trying to get rid of we're trying to get rid of our waste products with as little fluid loss as possible, right? So these two things are interrelated. And there is going to be an ability to selectively get rid of sodium or potassium or calcium. If we have too much of one of those, we don't have to get rid of all three of them at a high level. We can selectively tune through that selective secretion process within the tubule of the kidney. We can get rid of one or the other or all three or whatever we want, okay, in order to maintain the balance of each one of these concentrations within its own particular range. And finally, we can also adjust the pH, pH just being the proton concentration. Then finally, we need to get rid of our metabolic wastes like urea that we just talked about, creatinine, which is involved in muscle breakdown, and then any environmental toxin, any drug that you take, your kidney is going to filter it out. The rate at which it filters it out is related to the bioavailability of that drug and how long it's going to be functional within your body. A lot of the design of drugs is sort of wrapped up around making sure it can get through the first pass of the kidney and the first pass of the liver without breaking down, so it can get distributed to your body and act on the tissues where that drug is going to be useful. Okay, now what is the actual gross anatomy of the entire system? Okay, these are the excretory organs, at least in terms of the fluid excretory organs. We're not going to talk about the gut and digestion today. So we've got two kidneys. They're in the abdominal cavity here. The left one's a little bit higher than the right one, okay? And they are fed by a renal artery, and blood is returned by a renal vein. Now, these are very high pressure. The artery is very high pressure relative to any other sort of artery in the rest of your systemic circulation. It comes right off the aorta, okay? And it's going to get about 25% of the blood flow. It's going to go right to the kidneys. Okay. So you can imagine that that pressure might be intimately involved in how we're going to get enough fluid to come out into the kidney and get filtered through the kidney filtration process. Okay, so now moving down from those, we've got the ureters down here. These are ducts that have little muscles in them that can squeeze urine. Down into the bladder. And then we're going to hold the bladder, and we're going to hold urine within the bladder. So there's sphincters here. These are little muscular rings that can allow or block urine from traveling into the bladder. And we have sphincters here in the bladder itself. Okay, and that is, so, this is a sort of an evolutionarily conserved process where we don't want to just go dribbling urine around on us everywhere, leaving a trail of breadcrumbs for predators to find us. Nope. What we're going to do is we're going to store it up and release it only in safe locations. Okay, or if you're like a dog, you're going to go around and smell for somebody else's urine, and then you're going to pee on that spot and come back a day later and see if anybody else has been there. Right? It's actually a mechanism of communication for a lot of animals. Not so much in humans, but maybe a way to avoid, if a human smells like pee, probably you want to avoid, in my experience. Okay, so here we go. So next stop, let's now look at the kidney itself. Okay, so there's two structures. Okay, the kidney is basically the overall level of the kidney has got this sort of shape that looks like a giant bean. Okay, and you've got an outer rind to that bean called the cortex, and an inner zone of the bean here is called the medulla. Okay, so the term cortex and medulla we're going to hear multiple times. It's also on the brain. There's the cortex outer rind of the brain, and the medulla, something deeper inside the brain. And the adrenal gland is on top of the kidney. You're going to also have cortex and medulla. So that's just a common sort of terminology you should know. So we've got this cortex. Okay, and then medulla, and then embedded in these sort of parallel strands here are going to be things called nephrons. And the nephron is the individual processing unit of the kidney, okay? About 80% of the nephrons look like this. They have these short little loops, and about 20% of the nephrons are going to have these much longer loops. Now, the sort of main part of the nephron here, which is fed, each nephron has its own arteriole that brings blood into it and takes blood out of it, okay? An afferent and efferent arteriole. And each one of them has these little loops that distend down. Now, the main tubule zone of the cortical nephrons, the ones that basically live completely in the cortex, is up here in a higher layer. And then these juxtamedullary nephrons, that is, these nephrons with these long loops, called the loops of Henle, are going to sit, their main body, though, is going to sit down next to the medulla. Not in the medulla, but next to it. Okay. And that's because within the cortex here, the overall concentration of the sort of osmotic strength of this interstitial fluid is the same as the concentration in the blood. But as you go down into the medulla, there's going to be an increasing gradient of solute concentration till you get down from, say, 300 milliosmoles up here down to 1200 milliosmoles down here. Okay? And that gradient is going to be very important in the ability to produce urine on this hyperconcentrated. Okay. So, and what do these long loops of Henle do? Those are going to be involved in establishing and maintaining that concentration gradient in the interstitial fluid of the medulla.
So the.
Yeah.
Mhm. So they are in one, what does the urine?
So I would say both of them are involved in the production of urine. So this sort of structure here, this is part of the tubule stuff. Okay, this is in both the cortical and the juxtamedullary nephrons are going to be making urine there. And then this loop here, which is only really present, only goes down deep in the juxtamedullary, that's going to be what's going to maintain this concentration gradient within the medulla, which is going to be responsible for the ability of the kidney to hyperconcentrate the urine. Okay. Now, the nephrons, this is our individual processing unit, right? You're going to have a tubular component and a vascular component. So the tubular component, that's where the fluid that leaks out, okay, that gets filtered out, gets collected, okay? And it's going to travel around this little tubular pathway. It's going to dip down, it's going to come back up. It's going to travel through a distal tubular pathway and finally drop down this collecting duct. It's going to merge the output of multiple nephrons and then send it down the ureter into the bladder. So wrapped around these, the tubules are peritubular capillaries. So these are capillary, a whole capillary network that is surrounding the proximal area, the distal area, and this loop. Okay. And that is going to allow the exchange of material between the blood and the interstitial fluid here, and from the interstitial fluid to the tubule, and back and forth. Okay. And the overall organization of this capillary network is actually important. Okay, so we're going to have blood coming in here through an efferent arteriole into this glomerulus, and then it's going to come out of efferent arteriole, and it's going to travel all through this network of capillaries until it exits right here at the sort of end of this loop. So this capillary network here is, then this area is called the vasorecta, okay? And this loop is called the loop of Henle. So we're gonna look into this in a little bit more detail in a second. Okay, so the peritubular capillaries are going to be where we secrete and reabsorb material from the blood itself, and then that is going to exchange with material within the tubules. So you're going to get this bidirectional exchange that is going to actually be dependent on what receptors and channels and pumps are expressed within the tubule itself. We're going to go into this in great detail in a second. Now, one of the important aspects of this structure here, the vasa recta, okay, which is the descending and ascending part of this peritubular capillary, is that when it's descending down from this sort of network, there's actually directionality of flow here. Blood is going to come down this capillary network, okay? It's going to pick up sodium chloride. It's going to get, the blood is going to get more and more concentrated, okay? It's going to lose water because it is going to be in equilibrium with whatever is in the interstitial fluid of the kidney. Now, when it comes back up this loop of Henle, okay, so on the ascending limb, the ascending vasa recta, okay, it is going to start losing sodium and gaining water, so that when you come out of this sort of vein that comes out of here, that is going to be, you're going to be back to basically an isotonic level, an equal concentration of solutes than what is normal, as to what is normally found in the blood. So the directionality of this is important, okay? It actually operates in a process called countercurrent exchange. So in countercurrent exchange, this is a concept, and oftentimes we talk about it in the cardiovascular system, but it works for any space, but it's the overall concept. How can you have a very efficient exchange process between two areas? Okay, so in this case, our area is the peritubular capillary, is the blood and the tubule itself. Okay, you can also think about this in terms of oxygen trying to, in one chamber, trying to get into another chamber that trying to equilibrate, like oxygen in the blood. So the concept here is if you have a concurrent flow, if you have material moving in bulk down two tubes that are going in the same direction, and you want to extract as much stuff from one of these tubes and get it into the other tube as possible, if they're both going in the same direction, the best that you can do is get a perfect mixing, an even mixing of both compounds by unit, or of that compound by unit. Right? So if we start at 100% of something here, 0% of something here, it's going to always be going down this concentration gradient. There's going to be a very high gradient here. So you're going to get a lot of oxygen or whatever you want to move into this area until the gradient disappears. When there's equal concentration on both sides, there's no gradient anymore. You're not going to have any net diffusion of your thing, in this case, oxygen. You're going to have an equal mix. So that can be okay if you want to evenly mix between them. But what if we want to maximally extract stuff, maximally extract our oxygen from this side? In that case, the best way to organize this is actually to have the bulk flow go in opposite directions. Okay, so that the tube that has a lot of oxygen or whatever you want in it, and we want to get rid of, extract all of it, you're going to have this one running in this direction, and then we're going to have the tube that wants to get that material out and move it into it is going to move in the opposite direction. So in the case of the loop of Henle, okay, we're going to have the tubule going one way, and the flow through the tubule going one direction. We're going to have the flow of blood coming in the opposite direction. So in this case, the initial gradient is not that high. There's actually a very constant, this is pretty concentrated here in the blood coming back this way. It's even more concentrated in the tubule going this way. But there is still going to be a gradient, so you're going to be moving material from diffusion is going to be passing this material from a higher concentration to a lower concentration consistently all along the length of these two parallel passages. Because if the concentration is basically zero when it's coming in, even if you're just a little bit above zero, you're still going to get some diffusion down here. Okay, over here, you're maybe at 25%, and you're at 20% diffusion down here. Here you'll be at 75% and 70% diffusion down here. Here you're at 100%, 95%, you're still getting flow. You're still getting a gradient that's flowing down here. So countercurrent exchange is a way that you can maximize the efficiency of an exchange between two compartments that you want things to move down. Now, if we look in terms of the overall basic explanation of what the countercurrent exchange is going to do in the kidney, or within the nephron of the kidney, we're going to have the loop of Henle. That's the thing that dips down. This is going to create a concentration gradient within the kidney. The descending limb of the loop of Henle, we're going to have water leaving and filtrate getting concentrated. On the ascending limb, solutes like sodium chloride are going to leave, and the filtrate is going to get more diluted. And then the vasa recta is going to be moving in the opposite direction. It's going to have blood flowing in the opposite direction. That's going to get concentrated in one way and then diluted back again, back to its normal level by the time it exits. And then finally, to concentrate our urine, we're going to use the gradient established by the loop of Henle to reabsorb water selectively. Okay, so that's sort of a very sort of bulk overview, and we can then zoom in now to how all works. This is a pretty complicated diagram. I don't know if I want to walk through it all right now, but this is all in one place. We've got a vascular component that we're talking about, right? Those are the peritubular capillaries. What are the vascular components? We've got afferent arterial, so blood is coming in from our artery, right? Comes in, each nephron has its own afferent arterial, comes into this thing called the glomerulus, which is surrounded by Bowman's capsule, which is where the tubule starts and wraps around this. There's going to be a lot of leakage of fluids from the glomerulus here out into Bowman's capsule. And then it's going to exit the efferent arterial and then move into this peritubular network of capillaries. So afferent and efferent, these are also words that are going to be very common. You use them in neuroscience as well, right? Afferent just means something that's bringing in material to an area. Efferent means the stuff that's the part that's moving material away from that location. So if you are sort of talking about something inside the brain, some nucleus inside the brain, information that's coming in is from afferent projections into it. And when information is moving out of that part of the brain, it's an efferent signal. Okay, so this is now going to move into these peritubular capillaries, which are very sort of, concentrated and like complicated, and they're all wrapping around all these tubules. And there's going to be a lot of exchange of material between these tubules and the capillaries. But one thing to note here is that as this descends into the vasa recta, we can see that the overall net flow of blood is going to be in the opposite direction of the flow of the tubular fluid. Okay, so now to demonstrate that, let's look at our tubular component here. We've got the Bowman's capsule. It's going to collect the fluid. It's going to travel through this, what's known as the proximal tubule here. In the proximal tubule, we're going to get a lot of reabsorption of different ions, and then selective and secretion of selective secretion of stuff from the blood into the tubule. Then we're going to drop down into the loop of Henle. There we're going to have, this is going to establish our osmotic gradient. Fluid's going to come back up into the distal tubule. We're going to have some fine tuning of the concentration of things within there. And then finally, the collecting duct, where we will change the ability of the collecting duct to allow water to pass or not pass in order to adjust the overall concentration of the urine and make it hyperosmotic or not. Okay. That is where the filtrate is going to begin. And we're going to talk about that in just a second. But this is just sort of a summary slide that's going to give you your sort of overall overview of what each one of these sort of micro anatomical components do. Okay. One other thing to mention here is the juxtaglomerular apparatus. Okay, where is that? Okay, it sits right here, okay, in between the glomerulus and the capsule and this tubule here. And that's going to be sensing what's inside the filtrate passing through here. And it will adjust, it will release different molecules to adjust and tune the amount of flow coming into the glomerulus, which then can adjust how much fluid and filtrate is being produced. Yes?
Less water than the ascending blood flow contributes to
The descending blood flow takes less water than
the blood that's rising out. In the water that the rising up blood donate to that.
I would say that the blood here is going to be normal concentration, okay? The deeper it goes, the more concentrated the blood will get. And then as it returns back up, it's going to get diluted again, okay? The concentration of the blood is always going to reflect the local concentration of the intracellular fluid, okay? And so if you had a design where this vein came out the bottom of the kidney, where it's super concentrated, okay, you will flush out all of your salts. You'll flush out all the solutes from the kidney because the blood will be hyper concentrated when it leaves. It's very important to have this pathway for the peritubular capillaries to go dip down and then come back up and exit in the cortex, because in the cortex, interstitial fluid is normal concentration. So the blood can get concentrated down and then it's going to get deconcentrated. It's going to come back as isotonic. So it will not wash out the salts. It's going to temporarily borrow the salts and solutes and urea are going to move into the blood here, and then they're going to come back out by the time they get to the top. We'll look at that in a little bit more detail. Okay, so this is the anatomy. I know it's kind of complicated. This slide in particular, I think is a little bit excessively complicated, but it's trying to give you a sense of, what does a nephron actually sort of look like, right? So now let's look at the actual function here. Okay, overview of urine formation. Okay, so here are the sort of terms. So it's basically, we have a tubule. We've got this area of high pressure blood coming in. You're going to get leakage of blood plasma, stuff within the plasma, or at least fluid within the plasma, and small molecules of plasma. It's going to leak out here in a step called filtration. That material will be called the filtrate as it moves through the tubule. And then we are going to reabsorb a lot of stuff into our peritubular capillaries. We are going to selectively secrete stuff into the filtrates, into the peritubular capillaries, into the tubule, and then finally we're going to squeeze the end product, which is urine, in the excretory tubule. Okay, so what do we excrete? It's just what we filter minus what we reabsorb plus what we secrete into the filtrate. Pretty straightforward. Okay, so now we are going to, so the peritubular capillaries are always going to sort of reflect what is in the material in the peritubular capillaries will tend to reflect what is in the interstitial fluid. The control over what gets into the interstitial fluid and what gets taken up by the interstitial fluid is dependent on what channels, transporters, and pumps are expressed at each stage of this tubule. Okay, so the tubule is where the control of the system is going to be, is where it's going to be affected, or where the effectors for that control are. Okay, so let's start at the beginning here. The urine production is going to begin at Bowman's capsule. Okay, so this is just an area that, like, so we have maybe, it's just like a balloon that's you sort of encapsulates this sort of protrudence in this sort of very vascularized, almost like a little, it's captured capillary bed. It's like a little capillary bed that's surrounded by a balloon that's going to let fluid leak out of this capillary bed in a way that's much higher leakage rate than most capillaries, and then get captured by Bowman's capsule. Okay. Now we're going to modify that as we go through these different tubules, these different components, and then exchange substances between the nephron, interstitial fluid, and blood. And then once we get once we leave the collecting duct, we're not going to do anything to the urine anymore. Okay, so the urine is the final composition of the urine when it leaves the collecting duct is not going to change by the time it makes it to the bladder or in the bladder. There's no excretion or exchanges of material in the bladder. It's just a holding zone. So in greater detail, what is each step going to do? The proximal tubule, this is maybe a good one for studying. Okay, the proximal tubule, what are we going to reabsorb? The reabsorption process is mostly non-selective. It's going to absorb water, sodium, chloride, bicarbonate, which is a buffer, potassium phosphate, all these different things. Glucose, we want to amino acids, small proteins. Why are we doing that? It's because all of those things are small enough to leak out of the glomerulus, okay, in a non-selective way. And we actually spend a lot of our energy and resources actually collecting those things from the environment. We like glucose, unless we have way too much glucose in our system, right? If maybe we ate too much candy, just drink like, 48-ounce Coca-Cola. Maybe we don't want that glucose in us anymore. But we're absorbing all of this. We've gathered all this material. We do not want to just urinate it out. Okay. We want to reabsorb it. Okay. So that is going to be, most of that stuff is going to get reabsorbed in the proximal tubule. If it's not reabsorbed, if there is, glucose coming out of your urine, that's a sign of diabetes. Okay. Diabetes mellitus is actually, it's the word for sweet urine. It was originally detected as a disease by the doctor tasting your urine to see if it tasted sweet. Okay. Yes, that's true. That was probably due to, it is related because they're trying to dilute that concentration of glucose that's in their urine. So that having high glucose levels, so they might need to excrete more fluids in order to dilute that down. Okay. So that's what we're taking it back up. Okay, but we also want to get rid of stuff. Generally, we have an excess quantity of hydrogen ions in our body. Okay, so we want to secrete hydrogen ions. We want to excrete creatinine, which is a protein sort of breakdown product. And we want to excrete any sort of toxic drug metabolites that are floating around at low concentrations with our blood. Okay, so those stuff is going to get dumped out selectively into the proximal tubule. Okay. Now, in the loop of Henle, okay, we're going to go in the loop of Henle in greater detail. Each one of these areas we're going to go in greater detail. Okay, the descending limb is going to have a different composition of channels and pumps on it than the ascending limb. Okay, and that is going to allow the descending limb to let water come in. We're going to have channels, water channels, aquaporins, which will allow water to come in. Sorry. Descending limb, water is coming out. It's going to allow water out of the descending limb of the loop of Henle, concentrating urine. Okay. And then the ascending limb, the sodium, chloride, and potassium are going to get pumped into the peritubular capillaries, pumped out of this ascending limb of the loop of Henle, and that will start to dilute the fluid going on down here. So if we're losing water on the way down, we're going to concentrate this, and if we are pumping out the sodium, chloride, and potassium, we're going to start decreasing the concentration of solutes in here on the way back up. Okay. Then in the distal tubule, we're going to reabsorb a few extra things. We're going to sort of fine-tune the composition of our filtrate. We're going to reabsorb sodium, calcium chloride, and then we are going to secrete generally potassium hydrogen. And then finally, the collecting duct is where we're not going to really change much of those other solutes, but we're going to change the overall osmolarity of that fluid by controlling how many aquaporin channels are on this collecting duct. We have a lot of aquaporin channels that will allow water to leak out of the collecting duct with this concentrated surroundings that will concentrate urine. If we remove aquaporin channels from the collecting duct, that will not allow water to exchange, and it'll keep the urine the same concentration as you move down into the bladder. Yeah. Where does it go? So the stuff that gets pumped out is going to move into the interstitial fluid and then rapidly exchange with the peritubular capillary. So the peritubular capillaries are going to be leaky, or the peritubular capillaries are leaky enough that they can rapidly exchange with the interstitial fluid. You can imagine them basically being you can almost imagine them as being one compartment in terms of they're going to reflect the local concentration. What's in the blood is going to reflect the local concentration of the ions and small molecules in the interstitial fluid. Plus, the blood, of course, is going to have some big proteins and red blood cells in it. Okay. Yeah.
What does that mean to secrete? Is that being added to the urine?
So what does a secreted mean? Secreted means being added to the filtrate. Okay. And that is generally going to be a process that is either active, like we're burning actual ATP to do it, or a process that is going to be driven by a concentration gradient of another molecule that is going to get co-transported and drive that secretion process. And we'll look at that in the transport epithelium slides in a second. Yeah. Why is it so slim? Well, different locations within the loop, but maybe as like the outside of, you're taking something that has a lot more surface area and volume, the cortex, and then each one of those descending things has to get down to an area it's like going down a pyramid, right? It's getting more and more narrow. The amount of space that you have to get down into the concentrated core of the kidney is less than the volume that you have outside. Okay, so you're going to have to make them thinner. And actually, the components, depending on where you are here, you're going to have the thick area is going to have a different composition of pumps and channels than the thin area. Okay. In ever increasing detail. Now let's look at step one, filtration. Okay, we've got our afferent arteriole, we've got our efferent arteriole. We've got a glomerulus and Bowman's capsule. Okay. We're going to have 3 steps to our urine formation. We're going to have glomerular filtration. This is where the fluid leaks out of the glomerulus. Okay. Then we're going to get our tubular reabsorption and our tubular secretion in this tubule. Okay. So why are we calling it filtration? It's because not everything leaks out of the blood. We're filtering it. We're letting the smallest things, the fluid, the water, and the smallest molecules leak out. Plasma proteins like albumin or many other large molecules, your red blood cells are not going to leak out. They're going to be retained and travel into the efferent arteriole and then into the peritubular capillary network. After it goes through that peritubular capillary network, eventually it's returned to our venous system. Now, the amount of plasma that leaks out of here is huge, right? We're talking like 50, 65 times of your total fluid of your plasma in your blood is filtered through this every day. Okay, so it's a huge process. Okay, so about 20% of the the plasma that comes into here on every pass is going to leak out. Okay, so this is actually, a very it's like a high volume system, and this is why it's going to be able to be so powerful in maintaining the solute concentrations within our blood, because we're constantly filtering. And it's a very active process. Then we're going to have, so we have 20% of the plasma's leaking out here, and then we're going to have the selective movements of ions into and out, so that we're going to actually retain almost all of the solutes, except for what we don't want to maintain, which is that nitrogenous waste, the protons and things like that, which are going to go out of our urine. So what is driving the flow from the glomerulus into Bowman's capsule? It has to be a pressure difference, right? Fluid is only going to flow in a bulk way down a pressure gradient. So what are those pressure gradients? Okay, there is, first and foremost, we have the hydrostatic pressure. Okay, so this is basically the pressure of the blood sort of fluid pushing on the walls of the afferent arteriole, pushing on the walls of this glomerulus and trying to, leak out. It's a high pressure. Remember, this is coming right from the aorta of the heart. So the blood flow coming in here is at a pretty high blood pressure, 55 millimeters of mercury. It's much higher than in the capillary networks anywhere else in the body. Now, there is also some pressure resisting the exiting of that fluid. And it comes in 2 forms. One is the hydrostatic pressure that is in this tubule, in Bowman's capsule. This is not just leaking out into the air, where the pressure would be 0 relative to atmospheric pressure. It's leaking out into an actual enclosed space, that tubule. So the more that leaks out, the more this tubule is going to be increasing its pressure, and it'll be pushing back. Okay. Now, since fluid is flowing down the tubule and out to the collecting duct, it doesn't push back completely, but it does resist that flow. So that is this P fluid here. So the overall sort of hydrostatic pressure within this fluid, this enclosed tubule, is about 15 millimeters of mercury equivalent. Okay. And then we have this weird one, right? The colloid pressure. Okay, so this is the colloid osmotic pressure. What is that again? It's the same thing that we talked about when we're looking at fluid flow in other capillary networks, which is the sort of the force that motivates the diffusion of water, okay, based on the relative concentration of stuff floating around or dissolved in this blood versus in this fluid. Okay, so all of those proteins, the large proteins that aren't leaking out here, provide an extra, they form what's known as a colloid, and water is going to want to balance the concentration of that colloid inside and outside. Since these big things can't leak out, water has to leak back in order to try to balance that out. The amount of equivalent pressure of that is about 30 millimeters of mercury. So overall, our hydrostatic pressure of in the glomerulus minus the colloid minus the fluid hydrostatic pressure in the tubule makes a net filtration pressure of about 10 millimeters of mercury. And so that is what is going to cause the overall net flow of fluid from the glomerulus into the tubule. Now, the higher this net filtration pressure there is, the more fluid that will leak into the tubule. You'll have a higher rate of leakage, right? And the more that you are leaking in the tubule, the faster you have to clear that tubule out, right? And the total amount of fluid that then gets leaked out over the course of the day is known as your glomerular filtration rate. Okay? So high glomerular filtration rate is good. It's a sign of healthy kidneys. If you have all your nephrons attacked on both sides, you're going to have a great level of kidney filtration. If you're like my dad, who got kidney cancer and had to cut out big tumors on both sides of his kidneys, he has much fewer nephrons left. So his glomerular filtration rate is maybe 30% of a normal one. Okay. And as long as you can maintain it above a particular level, you're not going to have significant health problems. But if you go below a critical threshold, you will not be able to filter sufficient fluid through your kidneys. You'll have kidney failure. You'll have to start building up your nitrogenous waste, and you're going to have to actually go and get dialysis and get strapped into, machines that will filter the blood for you. Okay, to remove that. It's not a great life.
So does the blood that goes into the afferent arterial, is that at 45 millimeters of mercury?
That's a great question. I think yes, that's probably correct. Because it's going to be, it has to be flowing down a gradient here, so it's probably around 45. Yeah, that makes sense. Okay, so let's zoom in here on the microstructure of this glomerulus. Okay, so we've got our arterioles here. Okay, we've got Bowman's capsule here. The inside of the Bowman's capsule and the inside of the tubule is called the lumen. Okay, and then along this sort of glomerulus, this sort of network of these capillary beds, they are covered, okay, in these cells called podocytes. Okay, these podocytes are called podocytes because they have little feet on them, and those feet can change their positions to open or close passageways for filtration to take place. Okay, so to be filtered, a substance is going to have to pass from the blood here through pores and endothelial cells of each glomerular capillary, through a little basement membrane, and finally through these filtration slits that are sitting between each foot of these podocytes. And that is an area where we can regulate the amount of fluid leakage by changing how open or closed those slits are. Another way that we can change the glomerular filtration rate is changing the amount of pressure that's coming in. So there are smooth muscles here that can constrict or relax the arteries and the arterioles that are flowing into each nephron. So what would happen if we constrict them? You're going to decrease the ability, you're going to increase the resistance to flow here, and you're going to locally decrease the pressure on the far side of this constriction, which will then decrease our overall blood pressure within the glomerulus and lead to a decreased glomerular filtration rate. Okay. So if we constrict here, we're going to reduce the flow in, we're going to reduce the filtration rate. If we do vasodilation here, so make it bigger, you're going to have the opposite effect. You're going to increase the filtration rate because you'll have an increased pressure within the glomerulus. So this vasodilation and vasoconstriction is able, there's a homeostatic feedback mechanism where if your GFR is too high, you're going to constrict the arterioles. If it's too low, you're going to vasodilate. And regardless of your overall blood pressure, from a pretty wide range, all the way from sort of normal up to hypertension, you will have a sort of fixed constant glomerular filtration rate because of that ability to control the pressure into each glomerulus. Now, if we have very low blood pressure, you can reduce your glomerular filtration rate, and that can be bad for your kidneys. Okay. How does that actually work? What are the signals here? How does this arteria decide to constrict or relax? Okay, well, it's going to happen through two processes: a myogenic response, that's something built into the smooth muscle itself, and then paracrine signals, which are signals released from cells that sit next to this and travel locally in order to regulate that vasodilation or vasoconstriction. So the myogenic responses, the ones that are built into the smooth muscle, okay, all the smooth muscle cells have this intrinsic property where they can, if they stretch, okay, they will tend to try to contract. Okay, they respond to stretching by increasing their constrictive power. So if you have high blood pressure, they'll tend to get stretched out, and then they'll try to counteract that by constricting. Okay, and that'll be part of that negative feedback mechanism. Likewise, if your systemic blood pressure falls, they're not going to sense that stretch anymore, and they're going to relax. They're going to try to open up to allow more blood flow in. So that's just something that's built in, intrinsic to the smooth muscle around that area. But then there's also the paracrine signals that are released from the macula densa, which is this little area of cells that is affiliated with the juxtaglomerular apparatus, which sits sort of right here at the base of this glomerulus, and is also touching the proximal tubule. So if we have a high glomerular filtration rate, we're going to have fluid quickly moving through here. And then if the fluid is moving quickly through there, it doesn't have as much time to reabsorb sodium and chloride. And so the concentration of sodium and chloride here, when it reaches the macula densa, is going to be higher than normal. That is going to be sensed by those cells, and they are going to release signaling molecules, paracrine signaling molecules like ATP or adenosine, which then will travel over here to the smooth muscle and cause it to constrict. It'll trigger constriction. That will then reduce the blood flow into here, reduce the overall glomerular filtration rate, which will give more time for the sodium and chloride to get taken back up. By the time you get back out here, you're going to have a drop in sodium chloride levels, and so then it will stop the release of that paracrine.
So it's like if it's too much concentration, it restricts it, so it does more filtration.
If it's too much concentration, it's going to restrict it, so it gets less filtration and gives more time. So you have a slower flow here. You'll have more time to reabsorb the sodium and chloride on the way to this distal tubule. Okay. Okay, any questions about this? All right. So now let's turn to how does this actual selective reabsorption or selective secretion and reabsorption work? Okay, so we want to, balance here the amount of water that we're taking up and the amount of water that we're losing. We want to get rid of our toxic byproducts, and we want to be able to choose what molecules are going to get secreted into the tubule, and we will also want to be able to take up many of the molecules to be reabsorbed. Okay. This is going to be mediated by transport epithelia. So transport epithelia are really important within the kidney for managing that process of selective secretion and reabsorption. They're also super important in the gut. That's another area where you're going to be taking up a lot of, important, nutrients. So you have your gut is lined with transport epithelia. And they're going to have a couple of they're going to have a couple of features that are going to help them accomplish their function. One, they're going to have tight junctions next to each cell. That's going to block the transport of materials in between the cells. You're going to have a selective permeable tissue barrier, and you're going to have a very large surface area on one end of the cells. And then depending on what's expressed within the cells, plasma membranes on both the inside and the outside sort of end of these cells, you're going to be able to choose what gets in and out. So let's sort of look at the overall structure of a transport epithelial cell and how it relates to its neighboring cells. Okay, so first let's sort of get oriented here. Okay, so this is what's known as the apical side, the side that's facing the lumen of the tubule. So where all the tubular fluid is, okay, there on the outside, okay. And then we have the basolateral side down here, okay. And so on the sort of tubular, on the apical side, we've got all these fingers that are known as microvilli. What are they there for? They are there to increase our surface to volume ratio, right? We want to, instead of having just one flat sheet here, where it might limit the amount of transporters and things that we can put in there, or limit the amount of surface area that material can diffuse through. Instead, we have all these little fingers sticking out. We're going to dramatically increase our surface area. So that's going to dramatically increase our ability to move material across that plasma membrane. Another feature here is that there are going to be very tight junctions between neighboring cells. So here's an electron micrograph. Okay, this is like half a micron scale here, and you can see like these are nanometers tight. Okay, very tight, makes it very difficult for any small molecule, or certainly any large molecule, but even small molecules to move in between the cells. Okay, so this is what we're seeing here. Now, you could have some passage, this is called a paracellular transport, or it moves between the cells, but that is dramatically reduced in places like the transport epithelia from these tight junctions. Okay, it really is going to slow down diffusion, even of small molecules. So what's the consequence of that? If we're basically now packing the cells so close together, pulling them so tight that it makes it really hard for a molecule to diffuse from one end to the other end on the sides of it, that means anything that's going to travel between the lumen of the tubule and interstitial fluid and ultimately the peritubular capillaries is going to have to go through the cell itself. So that's going to be a transcellular transportation. What else do we have here? We've got desmosomes. Okay, these are areas that are going to also allow, like basically grab onto the other cells and allow structural integrity, like a network. So these cells are all working together as a giant sort of surface. And then finally, we're going to have some gap junctions. Gap junctions will allow ions, water and ions, to pass between neighboring cells. So they're going to again work as sort of a uniform surface, combined sort of a layer of material. And you can see the gap junctions here. These are little connections down here. This is sort of further down than the tight junctions. So they're going to allow passage between one to its neighbors of ions and stuff like that. Okay. Now, let's see exactly how this is going to work. Pretty complicated slide, pretty important slide. Okay, and this probably will. So let's look. Again, we have our lumen of our proximal tubule. Okay, so this is what's going on in the proximal tubule. Proximal tubule is where we're getting some non-selective reabsorption, and we're getting selective secretion of material. We have our lumen, that's the inside, this is the stuff, the filtrate that just came out up there. We've got our tight junctions here, so we're not allowing any paracellular transportation. Everything's going to have to be transcellular through here. We've got our little microvilli sticking out to increase our surface area. We've got our interstitial fluid down here, and we've got a peritubular capillary. The peritubular capillary will always reflect what is in the interstitial fluid. Doesn't have any sort of selectivity here. But how are we going to move material from the lumen into our peritubular capillary, into interstitial fluid? And how are we going to actually move stuff from here back into the tubule itself? All of this is going to be driven by the sodium and potassium pump. This is the most important one. Okay? This is where the energy comes from. You're going to take ATP, you're going to turn it into ADP, you're going to pump sodium ions out, you're going to pump potassium ions in. What will that do to the concentration of sodium in here relative to the concentration of sodium here? It's going to concentrate sodium here, it's going to deplete sodium from inside the cell. Likewise, it's going to deplete potassium here and increase potassium concentrations inside the cell. Okay. That's cool. Okay. So if we have a high concentration of potassium here, and we want to excrete that potassium into the tubule, how will we get it into the tubule? Do we need another pump over here? No. All we need to do is we need to just put an ion channel that selectively passes potassium. Because if we have a high concentration of potassium here, and a lower concentration of potassium up in the tubule, right? Because we're establishing a very high concentration of potassium in the cell now from this pump, it will just flow down this concentration gradient. You don't have to spend any other energy on it. It is going to passively diffuse, okay, into the lumen. Now, we've also depleted sodium on the inside here. So we have less sodium on the inside here relative to the concentration of sodium in the tubule itself. Can we utilize, so how are we going to sort of get sodium, sort of reabsorb sodium? We're just going to also have an ion channel that will allow sodium to passively pass through. Okay. We have higher concentration out there, less in here. We have spores that are selected for sodium, but sodium will just flow in. But now, how are we going to get glucose, amino acids, chloride ions, all those things that we maybe want to reabsorb into our blood, things that we spend so much energy trying to collect from the environment? How do we get those back? Okay. Instead of having different pumps for all those different molecules, instead what we do is we utilize the concentration gradient of sodium that is established by this pump and put in these things called cotransporters, which will only move these molecules if they are bound to a transporter with a sodium gradient, where the sodium is greater on the outside than on the inside. And it's going to use the energy of that, the chemical energy of that concentration gradient of the sodium to move glucose or amino acids or chlorides selectively into the cell, co-transporting it with the sodium. So if we have a very high concentration gradient of sodium and potassium, of sodium between the inside and the outside here, and not much of a gradient between glucose or amino acids, we can actually concentrate the glucose and amino acids by utilizing that difference in the sodium concentration. Less sodium on the inside or on the outside allows the co-transport of these molecules in. Once they're in here, we don't have those co-transporters on the basolateral membrane. We just put a little channel here that allows passive diffusion through a pore that's selected for one of those things. Then finally, how are we going to allow water to flow? We can allow water to flow by just putting aquaporin channels. These are water selective channels, water selective pores on both membrane sides. And we can then regulate how much water gets reabsorbed or not reabsorbed by the expression of those channels. If you express them a lot, then you're going to get a lot of water transport. If you remove them, you're not going to get any water transport. Okay, so that is a pretty important slide, and I'll leave it there. Okay, if there are any questions about this slide or anything else before we wrap up. Yeah. Because there's some secretion in potassium, there will be selective transporters for things that we want to get out. Also, in this transport of the fuel. Yeah. Yeah, let me just address it. I think it's getting a little bit, so the sodium is part of a countercurrent exchange, right? So sodium constant, and we'll look at that in a little bit on Thursday. Okay. Hi.
Good question, like, so it is about the counter current flow.
Mhm.
And can we move that to that slide? Here,
We are gonna, hold on. Yeah.
well,
The countercurrent flow is, we're gonna do like literally 10 slides on this.
Okay.
So let's just hit it.
okay.
Hit it.
And
Okay.
like another thing is, I still don't get it.
Can I,
Sure.
you're gonna have